Hello, I want to wait more people to come in first. I want to do it on live today. It is day 100. Yes, you are welcome to tell me what you want me to add today. If I got this stuff, I will add this today. If not, maybe I will wait until I got into the new project. Only me to drink it is too boring. I think I have to invite more people to drink with me. Actually, I put water bears into another box. I didn't put any water bear into this sandbox because I'm quite sure they cannot survive in this sandbox. Water bear is actually quite sensitive to the water quality. It means if the water is too polluted, it will not survive. Are they alcoholic? I don't know. Yesterday I sprayed a spray of alcohol into the sandbox. Let's see today whether they survive. I can smell a very strong and disgusting smell. Okay, for newcomer, it is Microbial Sandbox. It is actually a project we have done 100 days. We add random stuff every day and try to grow microorganisms. Actually, we did add some mushroom into the sandbox, but I think a very long time ago, maybe a month or 2 months ago, maybe we can add mushroom again into the new sandbox. Okay, let's do it. Which part of the sandbox do you want to see, like the surface or the bottom? Okay, I just pipette some of the content of the surface of the sandbox to a slide, and I'm going to put a cover slip on it. Okay, now I'm using the magnification 40. You can already see some microorganisms swimming inside. What are those dark red? Oh, you mean this? It is actually water bubble. Yes, air bubble, sorry. It's air bubble. Okay, we can zoom in. Okay, now we are following a dividing paramecium. It is actually the whole sample is occupied by majority of paramecium. You can see it is dividing in the middle of. But you can see they are actually not very active. You can see they swim very slow. Maybe because they're affected by the water quality, they are not actually very healthy. What will happen if you eat that? You mean eat paramecium? Nothing will happen because it is not parasite. Once it arrives to your stomach, it will be digested. And you can see the water is floating. So that big bubble here, actually you can never see anything inside the bubble because this type of microorganism is quite sensitive to dryness. If there are no water, they will die. Okay, he is a ciliate trapped inside the air bubble, so it burst. I'm now using a 400 magnification. And you can see the ciliate in the middle have already died. It is not moving anymore. But you can still see bacteria swimming because there's still a little bit of water. They can still swim. But the water is not enough for the ciliate to swim, so it died. Okay, I go back to the low magnification first. Later, I will check the bottom of the sandbox to see whether there's difference between the surface of the sandbox and the bottom. I don't know. It depends on you. If you want me to add anything today, you can tell me. You can tell me in the comment because we will still look at the sandbox tomorrow, but I will not add new stuff into the sandbox tomorrow because it is today is the last day to add things. But actually, there are not many things living in the surface. Okay, I zoom in. Okay, you can see this paramecium swim inside. Swim near some organic debris. You can see there are many small circles inside the debris. That it is actually the mold spore. Do you remember we add a moldy strawberry inside the sandbox? So there are a lot of spores inside too. And actually, you can see there are many similar balls inside the paramecium. Actually, they engulf those spores to try to eat them, but I don't think it is easy for paramecium to digest fungal spores because fungal spores are quite rigid. They have a very rigid cell walls. It is not easy for them to digest actually. Are there any seeds? I need to look for it. You can see in the background there are many bacteria swimming. Sorry, I just try to clean the screen because it is dirty and it affects the magnification. Because I haven't diluted the samples for a long time, like a week. So actually, the samples is full of many organic waste and toxins. Actually, it really affects microorganisms. So you can see them; they are not actually moving very actively. Yes, this Paramecia is actually going to die because you never see a ciliate not moving like this. Okay, we look into another part of the samples. Okay, now let's try to look at the bottom of the samples. There are many things sink to the bottom. You can see the water quality under the bottom of the samples is much dirty. Like a mud water. I need to clean the microscope first because I accidentally spilled some on the microscope. Am I screening the samples under the low magnification? It is 40 magnification. But so far, I didn't see anything moving, actually. And I still see some, like Paramecium here. But also it is not moving anymore. Yes, this one is dead also. And actually it is very common, because why they die under the bottom of the sandbox, because the oxygen concentration is quite low under there, so it tends to live on the surface. Okay, you can see in the middle there is a segment of Paramecia. It is the dead body of Paramecia, but actually you can see many things moving inside. It is actually they are actually bacteria. They're trying to digest the dead body of the Paramecia. Yes, there's like hair. And actually, there are many bacteria gathered together to try to digest the dead body of the Paramecia. But apart from that, I cannot see anything living here. Just many bacteria. You can see many bacteria swimming in the background. It is also the dead body of Paramecia. It will eventually be digested by bacteria. I think it is a good timing. It seems the ecosystem of the sandbox is actually going to end, because most microorganisms have died, and it is a good timing for us to start a new project, actually. And in the middle, you can see a green matter. It is actually algae cell. By the way, I will not throw all of the content of the sandbox away. I will keep some of them, and maybe I store it into a small jar, and we can look at this maybe after a long time. So it is just the end of the season 1. We have a season 2. We are going to have a season 2, so no worry. And apart from the sandbox, actually I have something to do today. Do you guys remember previously I tried to build a boss and try to grow water bear. Maybe it is a good time for us to look at this. Actually, I put many things inside this box also. Maybe we can see some water bear. I don't know whether they grow inside. But at least I put at least 20 water bear inside. So I don't know whether they reproduce. I just want to know whether they grow and build a community. Okay, let's look into this. Okay, in the middle you can see something like a sun. This one. It is actually a tested amoeba. Yes, it is actually amoeba. Some amoeba do not have shell. They just a moving cell. But this type of amoeba, they will build them a home, a shell, so that they can live inside and move together with this shell. And sorry. And actually, this type of unless you see it moving, otherwise you can never tell whether it is alive or dead, because this shell is actually made of calcium. It really takes a long time to be degraded. So even they die, they will leave this shell for a long time, and you can still see it after they have died for a long time. Okay, let's go for it. We are going to look at the water bear. We can. Actually, I'm not quite sure whether we can see water bear today. Or maybe we can, because I have a moss sample. Maybe we can try to look at this later. Okay, in the middle you can see a very fast moving microorganism. It is Halteria, a jumping ciliate. You can see it like jumping instead of swimming. Yes, this one, it is Halteria. And also some ciliate swimming. Again, I'm not quite sure whether we can see water bear today because I really don't know whether they will be produced inside the box. It is an egg, a microscopic egg, but I don't think it is come from water bear. It is maybe a water fur egg. A very small one. I'll put another slide. Maybe there will be more microorganisms here. This Euprote is also a type of ciliate. It is the one I said it looks like a cockroach. But it swims very fast. It is Euplotes. Euplotes. Yes, they use this method to try to draw some food inside the body, and if it is too large, or maybe they don't want it, it will filter out. It is just moving it cilia to draw water inside, and they will filter some food inside the body, and other unwanted waste will be filtered out. Okay, you can see in the middle, it is a microorganism, looks like a sun. It is actually called Heliosa. You can see it doesn't. It looks like it doesn't move, but actually it is moving. It moves very slow, and you can see there are many needle-like things radiating outside. Actually, it can be used to paralyze other microorganisms, and after paralyzing, it will engulf the microbes. And actually can see many things inside its body. It seems it has already eaten a lot of things. By the way, I still haven't added anything inside the samples. If you have anything you want me to add, you can tell me. And actually, I want to ask, what do you think is the best time to hold the live session? Because I don't know whether now is the good time. Maybe most of you have slept or go to work, go to school. Hello. We just finished checking the samples. Now we are looking for water bear, but it seems I didn't see anything here. There are actually many microorganisms swimming. You can see there are many things swimming around, but I cannot see anything looks like water bear. Can you see in the middle there's a hairy stuff? It is actually a leg of some insect. I don't know what insect it is. I think it will not be cockroach. Cockroach will not be so small like this. Okay, I check one more slide, and if there's still nothing, we will move on to the next part. I just forget we still haven't looked at the results of the WD-40. By the way, I'm planning to start the new project at around maybe Saturday. Maybe Saturday is better to start the new project. And also, I want to start making the pocket desk I mentioned before. Like, whenever we see a new microorganism, we can mark down to the pocket desk. In the middle, you can see a nematode, but it is not moving, maybe already died. It is also a nematode. You can see the size are quite different. This one is very large, and this one maybe a baby nematode. Why did everything die? Actually, like, microorganisms actually do not have a very long lifespan. They die very soon, but whenever they die, the body may remain in the environment for a period of time until bacteria degrade them. And also, I just said maybe it is die, but it is also possible that it is not die, because when it is not moving, it is not necessarily it is die. maybe, yes, sometimes they just, I don't know why they don't move, but after a period of time, they move again. The liquid, the liquid nitrogen, yes, I think it will kill all the things. But also it is not easy for me to get liquid nitrogen. So maybe, I don't know whether I will get a chance to add liquid nitrogen, but I want to try actually. And also remember, most microorganisms cannot tolerate a freezing, because freezing is a process that can disrupt cell membrane. So most microorganisms will burst, but some microorganisms like bacteria, they have cell wall. So during the process of freezing, the cell wall can protect bacteria from bursting. So after freezing, most surviving microorganisms are bacteria, but other things like ciliates and other microbes will die. And also, water bear can tolerate freezing because they can go into a dormant state. It allows it to tolerate freezing too. And actually, looking at microorganisms is a very time-consuming process, because you cannot just rely on a single slide to determine what really survive inside your samples. At least you have to take 5 slides, because the distribution is random. Maybe they gather at some points, but you didn't take it, you cannot see anything in your first slide. And in the second slide, you take another point, maybe you can see more microorganisms. So you cannot just rely on one single slide to tell you whether there are microorganisms. You have to take more slides to make the conclusion. Okay, at least there are some living nematode. This one, it is very active. It is not easy to focus because it is moving very active. Remember, most nematodes is harmless to human. They are not parasites. Although some type of nematode is parasite, but most species are not parasites. So even you drink it, you eat this, you will not get infected. I just thought it is water bear, but it is not. Okay. If any of you see something like water bear, you can tell me because it is also very easy for me to miss them. But so far we can just see many nematodes. We cannot. I cannot find any water bear. Hey, I found a water bear. But again, I don't know it is alive or die because sometimes water bear remain silent for a long time. Yes, it doesn't. It seems it doesn't move, but maybe you put here for like an hour, it will move again. By the way, it is from moss sample. It is not from my boss. So I really don't know whether it is alive or dead. But as you can see, there are many bacteria surrounding it. At least the water quality is not that good, actually. No, I seldom see water bear very active. Most of them walk very slow. Maybe, I don't know. But it really looks like they are digesting it, the water bear, because they gather around the mouth of the water bear. By the way, can you see a black spot inside the bacteria? If you pay attention to it, do you see there's a black spot inside the bacteria? the black spot actually is spore. Some bacteria can actually produce spore, so that when you kill them, like hitting it to, just like if you hit them or cook them, they will still survive because they have spore. Unless you try to boil them for like a few minutes, they will die. But if the temperature is not too high, like 60 degrees Celsius, it will not kill the spore. Can I show some microbes only visible in black light? But I don't have black light on my hand, so far I cannot show it to you. Actually, I want to capture this, but I think it is not possible when we are doing live, right? Anyway, I will look into this again. Sorry. Yes, it is maybe a dead water bear, and you can see there are many bacteria surrounding around the water bear's mouth. Maybe it is trying to digest the water bear. And actually, the skin of water bear is quite tough. It is not easy to be digested. But bacteria can digest water bear from inside the body of the water bear. Okay, another water bear here. It is definitely different species of water bear. You can see the skin color is different. This one is red. And you can see, apart from the skin color, I mean the size is actually quite different compared to the water bear we just met. But I think it is not moving. Oh, you just bought a microscope. I suggest you can look at like polluted water. You can see many things in polluted water. Or any fresh water. And remember, you can never see anything inside the tap water. Maybe you find a pond or a lake, you can find many microorganisms inside. And dirty water tends to have more microorganisms. If it looks very clean, you may not see anything inside. you make your own microscope with laser and lens. How? If you just put a leaf directly under the microscope, actually you cannot see it very clear because leaf is quite thick. It blocks most of the light. You cannot see it very clear. Like here it is leaf from moss. I can show you and zoom in. If you just look into the single layer, you can see very clear. But if you look in the middle of the leaf, it is not clear because it is quite thick. You cannot really focus onto it. By the way, if you want me to add anything inside the samples, please leave your comment. And if I have this item, I will try to add this later. Okay, in the middle, can you see a worm, but it is not moving. I don't know if you really can see a worm here. Or maybe you try to use your imagination. It is actually mosquito larva. The larva of Chironomus. But again, it is not moving. Dye some slides. You mean now? Sorry. You want me to add a drop of dye here? I can try. What dye do you want me to add? What dye do you want me to add? I have, because I've made it dye, neutral or basic or let me see. I have crystal violet, Aldin, saffron, and some Violin blue. Yes, I have gram stain, but if I perform gram stain here, all the all the microorganisms will die. Maybe I can just add some saffron. I can add now. I try to add this on the slide. I have a saffron. I will add a drop near the slide and try to stain it. I just add some onto the slide. We can zoom it to the location I just add the style. Here. It is diffusing to the whole slide. In the middle, you can see a ball, maybe a cell. It is actually a cyst. Oh, really? It's more visible now. Okay. Actually, it can stain bacteria, but I don't think the bacteria are getting stained. Okay, in the middle you can see a seed. I don't know what it is because it has already burst, but at least it is a ciliate. But it take up some dye, and now it died. Or you want me to make it dry? In the middle, you can see something like a pill. Actually, it is also a fungal spore. I just remember I made many moldy stuff inside a box, so maybe we can add later, maybe season 2. Yes, I do have a cat. You can see this moss, it is taking up the dye also. So you can see the outer part is getting red. And I think maybe we can build the season 2 jar together. Maybe we can also arrange a live session, but it really depends on, I don't know what time is more convenient for most people. Yes, we cannot see anything moving. One of the reasons is that we just add dye to the slide. So you can see in the right hand side, it is all red because we have add dye to there. And in the middle, you can also see a cyst. But again, I cannot quite sure what cyst it is. I'm now using 400 magnification. Maybe we just spend few more minutes on this line and we can add stuff to the sandbox again. This is also a rotifer, but again, you can see it is not moving anymore. Most of the things inside this moss is not moving. I don't know why. Maybe it is too dirty. You can see many bacteria swimming. I just asked you what dye you want me to add. Actually, what dye you want me to add? You just want me to add neutral dye. But neutral dye sometimes is not easy to staying microorganism, especially bacteria. You mean the circle cyst I just zoom in? But trichoderma is a fungus. I don't think it is a cyst or fungus. Also, I just missed it. Where is the algae? But you are right. Actually, there are many algae swimming, or even some are not swimming, and you can see they are green in color. Also, algae. I have methylene blue. Yes. You want me to add methylene blue? But actually, methylene blue can also kill bacteria. I can try it now. I add a large drop. Okay, let's zoom into this ciliate. There's a ciliate swimming in the middle, inside the methylene blue. Okay, at least it is still swimming, it is not die. But it is not staying. It did not take up the dye. But most other material inside the methylene blue getting really take up the dye. Actually, it is not too much because it is less than half of the slide. So I think it is okay. I still have half of the slide is not stained. And in my experience, actually microorganisms are very clever. They will not swim. They will not swim inside the methylene blue. So you can see that they tend to live in the safe place. It did not swim inside the methylene blue area. Okay, maybe it is time for us to add stuff inside the samples. Maybe we can we can play tomorrow again. Because tomorrow is really the last day of the sandbox. It is day 100 plus 1. We will look into the results of the sandbox again. Oh, sorry, I just forgot we still haven't looked at the WD-40 results. You can see actually it formed a layer. WD-40 is organic, so they cannot be mixed with water, so it covers the surface. Even if it doesn't kill any microorganism, it actually prevents air to diffuse inside the water. So actually the water now has a very low concentration of oxygen. But most ciliates need oxygen to survive, so maybe most of them have already died. I need to be careful. I don't want to spill it all over the desk. I take both the surface and the bottom of the content and mix together and try to look under the microscope. Oh shit, I need to clean the desk. Yes, maybe I do it with. Actually, it is the boss we used to grow. I try to use it to grow water bear, but we just checked it. We didn't find any water bear inside, so maybe it failed. This is the slide we just looked at. The left side is saffron, the right hand side is methylene blue. We add different type of dye to the slide. Okay, now we are looking at the WD-40. so far I still haven't seen any moving microorganism, but I see many spore. I cannot do it with 1000 zoom because we are doing wet mount. Wet mount means we add a drop of water and cover it with a cover lid. But we can never use 1000 magnification to look at this because you will never see a clear picture. 100 objective is used for oil lens. You need to add a drop of oil on the slide to use the 100 objective to achieve 1000 magnification. It is not possible for me to buy an electron microscope unless I got funding. But so far I don't think it is possible for me to get funding to buy a electron microscope. Although I just received a microscope from a company and we need to test with it later, but not today. Yes, can you see a lot of circle, a lot of oval stuff here? Actually, it is mold spore. Because I take the samples near the moldy strawberry, so there are a lot of mold spore diffused into the samples. Hydrogen peroxide. I don't have pure hydrogen peroxide on my hands. I just have hydrogen peroxide mixed with isopropanol. See algae, but I don't see any algae. You mean the oval stuff all over the slide? It is actually spores, fungal spore, and in the middle there is a dead body of Paramecium. And at least we know WD-40 can actually kill everything. I really cannot see anything survive, but we can actually look at bacteria. We can see whether bacteria can still survive under WD-40. I need to change to phase contrast objective. And maybe I focus to another. You mean in my place? It is 12:43 a.m. I think I don't need to zoom in because I know I can still zoom in, but I think I don't need to use phase contrast microscope because I can see many bacteria swimming. Oh, we have just looked into the surface of the samples. Actually, nothing special, just some Paramecium. Actually, mostly Paramecium. I cannot see many other microorganisms. And at the bottom of the samples, there's nothing. Nothing living in the bottom. Actually, not. I'm quite sure my English is actually not very good. No, I'm not from Beijing. I'm not from China, actually. Okay, maybe we can start adding stuff to the samples. Wait me for a few minutes. Your comment get deleted. Do you have left any comment in my new video? I don't know because I never deleted any comment. Even root comment, I will still leave it there. I will never delete any comment. I'm not making joy. It is a jar of microorganisms. We have just looking into it, and maybe we can add more stuff. Let me see. Can you add some wood ash? I have some wood here. Do you want me to burn it and turn it into ash and add into the samples? I'm not from Mongolia. I think people from Mongolia cannot access YouTube. Okay. Okay, let me find the wood first. Okay, now my home is full of smoke and smell. Okay, next, I forgot to take video. I need to put it in my shot. Sorry, maybe we do it again. Add protein. What protein? You want me to add a meat or protein powder? Actually, I don't have protein powder now. Algae. Let me see, algae. Actually, I still got some algae from the day 44 too. Actually, I can still put it into a sandbox. Okay, let's add the algae inside. Spirulina. I still have some spirulina. I need to find. Yes, spirulina here, Korean brand. Oh shit. It got moist. It got moist. It formed a solid. Yes, like this white powder. I think I just add the whole pack inside. I don't want to keep it anymore. Add blood, maybe not today because I don't have a clean needle today. I need to buy a sterile needle because it needs to be done very carefully. Put 4 good suggestions. Put them in a pool to see what the viewers will pick. It depends on how to define a good comment. Does it mean I pick 4 comments that got most liked? What engine oil you mean? Isn't it just WD-40? It was no engine oil, but maybe next time because I cannot get engine oil now. I just find out that I got a moldy soil. I got a moldy soil. Do you think I still add inside the sandbox? Because sometimes I put the tube aside and I forgot to wash them, so it become moldy now. You want blood vs microbe? Maybe we can do it in the new season, but not now because I don't have a clean needle. Sorry, potato. Suit. Suit. The new season will come maybe Saturday. I will start the new season at Saturday. I don't have suit too. Maybe I can knock it down first. Maybe in the new season we can add this. I need to find whether we have potato, black powder from the carbon deposit. Actually, I can now burn a paper, and is it counted as soot? Put from my pet. because we just clean out that poop, so I don't have any poop now. Maybe, do you want fish poop? I have fish poop. I can just get the bottom of the fish tank to get some fish poop. Burn a candle and put a ceramic and glass plate above the flame and some black powder is formed. Burn a candle. Oh, okay. I get it. You mean this? Maybe I put the whole slide to Sandbox. I can also add again in the next season. I want to add some methylene blue. Capsaicin. Do you remember we just maybe 10 days before we do experiments with chili, I mix a part of the Sandbox with Thai chili, and they really kill most of the microorganisms. I don't know whether it is relatable. I don't know whether it's really proof that spicy stuff can really kill microorganisms. I still keep the bottle here. Maybe we can look again. This is the part of the Sandbox which we previously mixed with some chili. I don't think there are anything inside actually, although it looks turbid. You mean which one? You mean the jar? It is not lake water. Yeah, again, you can see there are no microorganisms swimming anymore. But you can see in the middle there are some plant cell from Chile. So you can see there's some red color inside the cell. Cut the seed and stuff inside. You mean I just put the skin of the chili? Yeah, I cannot see anything. So maybe we can try later. I still got some chili actually. But I don't have capsaicin. Okay, maybe. Can I just use the Thai chili? Or do you want a more spicy one? This is I'm looking at the chili skin, so you can see a piece of red color. Okay, maybe we can try it in the new season. Okay, maybe we finish. Okay, do you still have anything you want me to add, or we can stop here maybe? I think tomorrow I will still hold a live session to look at the last day of the sandbox, and the whole project will finish. What at what? Sorry. Oh, you just deleted the message. I don't have biochar. This suggestion is so specific. Sodium dichromate mixed with sulfuric acid. It need to be found in professional laboratory, maybe later. Fertile soil. Maybe I can find some later. What I got now is quite limited because it is in the midnight. Maybe we call an end here. because I still need to edit the new video. So before I sleep, I need to do it. So maybe I call an end here. And tomorrow, maybe I start the live session exactly the same time. I don't know, maybe around this time, yes. Tomorrow I will start the live session again, and we will look at the day 100 plus 1 of the samples. Acetic acid, okay. I can add it because I have acetic acid. But you want me to mix acetic acid with sodium dichromate or just add acetic acid to the samples? For your information, I'm not living in China. It is 1:30 in my place. I'm sorry, I just realized I don't have acetic acid. I just have actually they are similar. Acid. Let me see. I just have acetone alcohol. Would you mind? I said I'm not from China. Maybe I just add a little bit because it is my gram stain material. I don't want to waste it. Okay, anything you still want me to add? If not, maybe we just call an end here. Put candy and toss it with sour candy. Let me see. Let me see if there's any candy inside my home. Actually, glucose is a good idea. I think, yeah, maybe we can add to the new samples. Maybe I just call an end here. Actually, I really want to do a statistic about what what materials do we so far have added to the samples? Like how many of them and how many times did I just add them to the samples? But I don't have time to do this. I cannot tell you the exact time, but I think tomorrow, or do you want Friday? If you want, I can. We can wait just for Friday. And actually, Friday is more convenient for most people. And may I know actually what time do you think is most convenient? But actually tomorrow I'm not quite free. So if I really start the live session tomorrow, you need to wait very late. I cannot do the live session as early as today. If you want me to start the live session exactly the time, like today, maybe Friday is more convenient. Okay, then maybe I think maybe we can take a break tomorrow. I can publish the day 100 shorts tomorrow, and do 100 plus 1 day on Friday. Friday night. What do you mean Friday night? You mean which country? I mean, because many of you come from different countries, so I don't know. And actually, it is more convenient to communicate like using US time. Maybe you can tell me. Oh, Philippines. Okay, now I can see many people are from different country. If we cannot draw a conclusion, maybe we can start exactly the same time like today. Where I'm from, it is a secret. Hello everyone, what is happening? It is nearly the end of the live session. Thank you for subscription. Yes, I'm going to end now. So maybe we can start on Friday exactly the same time like today. Okay, see you. Bye bye. Oh, thank you very much. You have watched my video for a very long time. I really appreciate. Thank you. Oh, thank you. If you really can do the video like this, I can share your video. Okay. Okay. Good night, everyone. Okay. I see your comment. You have watched my video also for a long time. Thank you for your support. Thank you. Okay. Goodbye.